is an automatic DNA synthesizer, and what you can see here are the nucleotides A, G, C, and T. And so what this machine can do, we can program it to put in, as we're synthesizing the DNA, we can specifically put in A at this position, then a C at the next position, and a T and so on, and then thereby resynthesize a part of a gene. And so in essence, we have the capability of changing the genome in any way we see fit.
He then has another machine that incorporates the new piece of DNA into the mouse cell.
We put an enormous voltage jump on this, and in the process what happens is that the little holes are opened up in the cells, and the DNA slips into the cells and then goes into the nucleus and then participates in making the DNA of the mouse.
In this way, he can change the mouse embryo when it's just a small cluster of cells, and then observes how the new DNA affects the way the animal forms.
Will it affect the growth of cells? Will it affect making particular bones? Or will it affect making the muscle or the tendons in that limb? Or will it affect all of those?
This is one of the best ways to study what specific genes actually do. He sees how the new DNA affects the embryo, how it changes the adult mouse, even how it alters the mouse's offspring as the changes pass through generations. This mouse has had a gene switched off so it cannot move its face muscles. It cannot blink. Now that might sound cruel, but Mario Capecchi does this because there are children who are born without the ability to move their face muscles, and his research is designed to look for a cure.
The amount of knowledge that we get by studying the function of a gene in a living organism is enormous. I mean, most of our previous knowledge we were off completely as to what a gene is doing in a living organism. And the only way to find out is actually to alter it and see what happens. Genetically, mice and humans are almost identical. That is, 99.9% of all the genes in a mouse and a human are going to be the same. So whatever actually we learn in a mouse is going to be directly applicable to humans.
The technology that he's using to do this could have another application. Just as Dolly the sheep has opened the door to human cloning, Mario's mouse shows the way towards human enhancement. But if the technology were to be used in this way, would science have crossed the threshold of what's acceptable? Most agree that Mario's work should be used to cure illness, but many question trying to use it to make us more intelligent. However, Watson doesn't see a clear distinction between the two.
By saying healthy, your brain is also healthy, and if you really are stupid, I would call that a disease. Stupidity is, a disease of the brain. The lower 10% who really have difficulty, even in elementary school. What's the cause of it? A lot of people like to say, oh, it's, poverty and things like that, but it probably isn't. And so I'd like to get rid of, help that lower 10%.
Does Watson draw the line anywhere? How about more cosmetic changes, making people better looking?
Everyone was saying it would be terrible. We made all girls pretty. Okay. Well, I think it'd be great.
And it's those kind of comments that make some people think that Watson's judgment has gone off the rails. They argue it would be crazy to make such changes to our DNA, lumbering generations to come with our current cosmetic preferences. But before discounting his views, one should hear what else Capecchi is planning. He's turned his attention to chromosomes. Our DNA is wrapped up into 23 different sets, which carry our genetic traits from one generation to the next. Capecchi doesn't want to tamper with any of these. He's thinking about making an extra artificial chromosome. It would carry a bundle of genetic improvements, which he could switch on and off, just like he does in mice. The artificial chromosome could even be upgraded, like computer software, before being passed on to the next generation. Any alteration comes with a kind of money-back guarantee.
It does in the sense that you're not stuck with it. I mean, you're not changing the genome forever. You can change it, and if you're pleased with the results, you can keep it. If you don't like it, then you get rid of it and go back to the previous situation. And this is very important because what we may think is brilliant today, 20 years from now may seem pretty stupid. And whatever they think about in 20 years from now will be stupid 20 years hence. So it's very important to make the system reversible.